Hello everyone, I am Luciano, and I'm going to give you an overview about an ongoing project I'm working at the University of Bolzano here in North Italy, in association with the Bocconi University of Vienna. In this overview, I will show you just some preliminary data as I start working on this dataset not long ago, and there is still plenty of work to do. My work is about the role of genetic polymorphisms in the diapause behavior of Ipis typographus. Ipis typographus, also known as the European spruce bark beetle, is a terrible pest that has been causing serious damage on cultivated spruce trees all over Europe. Generally, Ipis typographus undergo diapause by the end of autumn and start emerging from this state during mid-spring to start a new generation. This condition is known as univoltine, or individuals that only have one generation per year. However, increases in the temperature and progressively warm springs due to climate change are relaxing the selective pressures on Ipis typographus. And what we can see is an increase in the frequency of multivoltine individuals, or individuals that have two or multiple generations per year. This outbreak reduced the time for spruce populations to recover from Ipis attack, which have already caused the loss of thousands of square kilometers of forest here in Europe. To better understand this phenomenon, my collaborators decided to investigate the mechanisms to regulate the diapause behavior on Ipis typographus and design a few experiments to understand them. On the first paper from 2017, they discovered that the diapause is triggered by a reduction on the day length, or photoperiod, but also that the triggering of the diapause can be overridden by a rise in temperature. On a second work from 2022, they explored the diapause on different European Ipis populations and discovered that they were structured in Northern European beetles exhibit obligate diapause, irrespective of the photoperiod, while Central European beetles display both obligate and facultative diapause. These studies were fundamental to understand the diapause behavior on Ipis typographus and to characterize it as a phenotype on different populations. But what about the genetic variation? So, to start understanding how the genetic variation of diapause occur in Ipis typographus, we designed an experiment to try to capture the extremes of the phenotypic variation. We collect about 600 beetles from 3 populations. One represents univoltine from North Europe, here in Sweden, and 2 from Central Europe, composed by uni and multivoltine individuals, tests in laboratory before DNA extraction. For the Central European populations, we collect also individuals occurring in low altitudes, close to 300 meters, and in high altitudes, close to the mountain tree line, on altitudes about 1,500 meters. For all individuals, we extract the DNA and generated a reduced genome representation with double digestion red-seq. And for the subsequent analysis, we will use the Ipis typographus genome available on GenBank. Now I will present you some of our results. First, we need to guarantee that the populations have sufficient genetic differentiation to be able to detect any possible trend among the populations. So I use the IPyRed pipeline to build the reduced genomic assembly, and then I remove all the samples with 15% or more missing data and use it to build a tree with the unique SNPs. As you can see here in the tree, the population clustered accordingly with their geographical localities, with north, high altitude, and low altitude, forming well-defined groups. univoltine and multivoltine individuals, however, did not cluster inside each respective group. Continuing with the genetic differentiation, we calculate the relatedness of each population using the entire SNPs matrix under the PCA analysis. Once again, the three populations cluster accordingly with their geographical locality, as you can see here in the figure. This analysis just complements the previous one, showing the populations were really different. Okay. Next, we start to explore the genome-wide association of the polymorphism to see if we could find associations with any particular trait of our populations. We test all possible pairs of our populations using all the SNPs. As you can see here, despite being many, over 1.5 million SNPs, only a few showed genomic-wide significance, those above the blue line. So we also decided to check the pairwise FST to quantify how genetically distinct the populations are from one another based on the frequencies of the alleles found. The results were kind of what we expected, with North European populations being more similar to high altitude populations than to low altitude populations. Our hypotheses predict that due to harsher conditions on high altitudes and high latitudes, the pressure of multivoltine individuals is higher than in the lower altitudes with softer conditions, which is more or less shown in the figure here. The differentiation, not the pressure. As the Central European populations come from geographically near places, this result suggests a strong influence of altitude on the genetic differentiation of Ips typographus. After we verify the effect of altitude on the loci frequencies, we decide to run some covariate tests to eliminate false positives. These analyses were run on bypass, and we test the significance of the SNPs when covariating the populations regarding the phenotype, diapause or not diapause, the geographical coordinates, latitude and longitude, and altitude, high and low altitudes. The results were interesting. We could reduce our significant SNP matrix to about 1,000 markers with statistical support. As you can see here in the figure, all the SNPs above the red line on each plot were considered relevant for the subsequent analysis. So with this reduced analysis, reduced number of SNPs that we confirm statistical support, we need to go after the biological meaning of this. And that's the part I'm currently working right now. So for the about 1,000 markers, I am manually curating them, checking in the genome where the markers occur to see if they hit any gene. If the SNP is a hit on an annotated region of the genome, I then go to the available data sets and check the functional annotation of this region. So here you have an example of the chromosome 1, and these small dots, sorry about the figure here, they are very small, but the small dots are the hits on the chromosome 1. So I'm going to see if these parts of the chromosome are annotated, and I check in the database for the functional annotation of this part of the genome. Our goal with this is to identify the functions, domains, or protein families that are described in the literature that have historically been associated with things like circadian rhythm, energetic metabolism, and biological cycles, which are known to have a strong influence in the diapause behavior in many other groups of insects. If we succeed, we'll be able to link the specific region of the Ipsis typographus genome to the unimultivoltine phenotype. This information can be readily used to monitor bark beetle populations here in Europe and to check for possible changes in allele frequencies within each population during their years, which can be used to predict and combat future outbreaks of this species. Thank you very much for listening.